Hello friends, welcome to my channel. We were studying light reaction in photosynthesis. Welcome to part 3 of light reaction. So first of all, let us study the structure of chloroplast. Chloroplast, it is the green colored pigment which is present inside the mesophyll cells of the leaves, and it conducts the photosynthesis. Light reaction and dark reaction occurs inside the chloroplast. So the chloroplast is a double layered membrane. This is the outer membrane of the chloroplast, and this is the inner membrane. This is the outer membrane and this is the inner membrane, and inside the chloroplast there is thylakoid. These are the thylakoids and these are arranged one above the other. So these are known as, this complete structure, it is known as granule thylakoid. This is known as granule thylakoid. This complete structure is known as granule thylakoid. These are thylakoids, stacks of thylakoids one above the other. Two granule thylakoid, they are connected by a connection which is known as stromal lamellae. This is known as stromal lamellae. And this complete area, it is known as stroma. This area, this matrix, it is known as stroma. Once again, let us discuss. This chloroplast is a double layered membrane. The outer layer is called as outer membrane, inner membrane. Inside this, there are many thylakoids. Thylakoids are present as stacks one above the other, and this complete structure is known as granule thylakoid. Two granule thylakoids are connected together by stromal lamellae. So in the last class we have studied photosystem. Photosystem 2 and photosystem 1, it is present on the membrane of the granule thylakoid. On the membrane of the granule thylakoid, photosystem 2 and photosystem 1 is present. But in the stromal lamella, we can see only photosystem 1. Once again, I will tell you, there are two types of photosystem, photosystem 2 and photosystem 1. Photosystem 2 and photosystem 1 are present on the membrane of the granule thylakoid, and photosystem 1 is present on the stromal lamella. I hope you have understood the structure of chloroplast, which will be helpful in studying light reaction and dark reaction further in photosynthesis. Structure of photosystem. In the last class itself we have seen. Let us revise once more. A photosystem, it consists of many pigments. In one pigment it absorbs light energy and the electron is excited, and the electron is transferred to or given to another pigment. While the transport of electron is given to another pigment which has higher wavelength and less energy. And finally the electron, it is given to reaction center. Reaction center is chlorophyll A. Chlorophyll A is called the reaction center. It is the main pigment, and the reaction center, it gives the electron to primary electron acceptor. There is a primary electron acceptor in the photosystem. It takes the electron. Apart from it, there are enzymes in the photosystem. Since the reaction center, it has enzymes attached to it, it can carry out photolysis of water. Photolysis of water is breaking of water to give hydrogen ions, electrons, and also oxygen. So once again we will study the photosystem. Photosystem consists of many pigments. These pigments all together, it is known as antennae, and it consists of chlorophyll B, xanthophyll and carotenoids. Chlorophyll B, xanthophyll and carotenoids. The electron will pass on from a pigment which will absorb light energy at higher wavelength from a pigment which absorbs light energy at lower wavelength, and the energy will decrease when electron passes from one pigment to another pigment. Finally, it is given to the reaction center. From the reaction center, electron is given to the primary electron acceptor, and enzymes are also present in the photosystem. So this is the reaction center, it is chlorophyll A. This is called the antenna, which consists of carotenoids, xanthophyll and chlorophyll B. And this is the primary electron acceptor, which accepts the electron. It has to take the electron to get the work done. Now enzymes are also present in the reaction center or attached to the photosystem. That's why reaction center, it can carry out the photolysis of water. Photolysis is breaking of water to give hydrogen ions, oxygen and also electrons. Let us see the photosystem. There are two photosystems in the light reaction. There are two photosystems, that is photosystem 2 and photosystem 1. So there are two photosystems, photosystem 2 and photosystem 1. Photosystem 1 was discovered first. Photosystem 1, it is named as such because it was discovered first, and the photosystem 2, it was discovered second. In photosystem 1, in the chlorophyll A, that is the reaction center, it works at 700 nanometer. In photosystem 1, chlorophyll A, that is the reaction center, it works at 700 nanometer. It works at a wavelength of light 700 nanometer, and photosystem 2, it works at 680 nanometer. So can you understand the difference? Photosystem 2, it works at 680 nanometer, and photosystem 1, here the chlorophyll, it works at chlorophyll A, it works at 700 nanometer. PS1, photosystem 1 is said so because it was discovered first, and photosystem 2, it was discovered second. It has a reaction center, and these are the pigments. These are the pigments. This is the main reaction center. This is the enzyme. Here this is the enzyme in photosystem 2, and this is the primary electron acceptor in photosystem 2, and this is the primary electron acceptor in photosystem 1. The primary electron acceptor in photosystem 2 is called pheophytin. The primary electron acceptor in photosystem 2, it is called pheophytin. The primary electron acceptor in photosystem 1, it is called iron sulfide, FeS. FeS. The primary electron acceptor in photosystem 1 is iron sulfide, and the primary electron acceptor in photosystem 2, it is pheophytin. And both the photosystem, it become active when light falls. And this photosystem 2, it can function only at 680 nanometer, and photosystem 1, it functions at 700 nanometer. Hi friends, let us study light reaction. Please listen carefully. In the previous, I already have told you that where the light reaction is taking place. Light reaction is taking place inside the chloroplast. So inside the granule membrane of the granule thylakoid, this is the membrane of granule thylakoid. This is the membrane of granule thylakoid. This is the membrane of granule thylakoid. In the membrane of granule thylakoid, photosystem 1 and photosystem 2 is located. So here is where the light reaction is taking place. This is the lumen or the empty space. Lumen is nothing but the empty space of granule thylakoid. This is the lumen, and on the thylakoid membrane, this light reaction is taking place. On the thylakoid membrane, photosystem 2 and photosystem 1 are present. And this is the empty space. This is inside the lumen of the granule thylakoid. Now I have zoomed this diagram like this. I have made a big picture of this diagram. Here you can see this is the membrane of the granule thylakoid. This is photosystem 2 and this is photosystem 1. When light falls on this photosystem, on both the when light falls, both the photosystem 2 and photosystem 1, it becomes active and the electron is excited. Electron is excited and as a result energy is released. When light falls on both the photosystem, electron, it gets excited. For example, if this is the wavelength of light, if light is less than 680 nanometer, both the photosystems will get excited. Light should be less than 680 nanometer, 680 or less than 680 nanometer. The wavelength of light, it should be less than 680 nanometer. If the wavelength of light is more than 680 nanometer, then PS2 will not work. The wavelength of light should be less than 680 nanometer or equivalent to 680 nanometer, then only PS2 will work. So both the photosystem, when the wavelength of light is 680 nanometer or less than 680 nanometer, both the photosystem, it will become active and the electron is excited. Either the electron is given back to photosystem. If it is not given back, the electron is accepted by the primary electron acceptor. What is the primary electron acceptor of PS2? Pheophytin. And the primary electron acceptor of PS1 is FES. The electron, it is given to the primary electron acceptor and energy is released. So when light falls on the photosystem, when light falls, both the photosystem, it becomes active and the electron is released. Electron is given to the reaction center, and from the reaction center, the electron moves to the primary electron acceptor. In PS2, the primary electron acceptor is pheophytin. In PS1, the primary electron acceptor is iron sulfide. Hope you understood this much. Please watch the video again if you have not understood. Wavelength of light is more than 680 nanometer, PS2 will not function. Wavelength of the light should be equal to or less than 680 nanometer, then only PS2 will become active. Whenever the electron gets excited in PS2, it is given to pheophytin, which is the primary electron acceptor. From pheophytin, the electron, it is given to plastoquinone. From plastoquinone, the electron is given to cytochrome B6F. And from the cytochrome B6F, the electron is given to plastocyanin. And from plastocyanin, the electron is given to PS1. And from PS1, the electron is given to iron sulfide. Iron sulfide is the primary electron acceptor of PS1. The iron sulfide, it gives the electron to ferrodexin. And ferrodexin, it gives the electron to NADP+. NADP+, it accepts the electron, and this is known as reduction reaction, and it forms NADPH. And NADPH, it is formed in the stroma. NADPH is formed in the stroma. Once again we will study. When light falls on the photosystem, both the photosystem, it becomes active, it releases electron. The electron reaches the reaction center. From PS1, the electron is given to primary electron acceptor, which is pheophytin. Then the electron is given to the electron carriers. These are the electron carriers. The electron is first given to plastoquinone, and then it is given to cytochrome, and then cytochrome B6F, and then it is given to plastocyanin. And the electron, whenever the electron moves from one pigment to another, energy reduces. So electron is said to travel downhill. This is the electron transport chain. It consists of plastoquinone, cytochrome B6F and plastocyanin. The electron is said to travel downhill. Energy is downhill because energy decreases when electron moves from one pigment to another pigment. Then finally the electron is given to PS1. PS1 gives its electron to the primary electron acceptor, which is iron sulfide. Iron sulfide gives the electron to ferrodexin, and ferrodexin gives the electron to NADP+. NADP+ accepts the electron because it is already electron deficient, and it forms NADPH. So this is a reduction reaction. Whenever a compound, it accepts electron, it becomes reduced. Whenever a compound, it accepts electron, it becomes reduced, and whenever a compound, it gives electron, it becomes oxidized. So we have seen the electron transport chain. Electron transport chain consists of 3 electron carriers: plastoquinone, cytochrome B6F, and then plastocyanin. And the electron always travels downhill in the electron transport chain because energy is getting decreased when electron moves from photosystem to electron. When it gets excited, it gives the electron to primary electron acceptor, which is pheophytin. Then the electron, it moves across the electron transport chain, which consists of 3 electron carriers: plastoquinone, cytochrome B6F and plastocyanin. Electron is given to plastoquinone, then cytochrome B6F and then plastocyanin, and finally the electron is given to PS1. PS1, it gives its electron to iron sulfide, which is the primary electron acceptor. Iron sulfide gives the electron to ferrodexin, which is a protein. Ferrodexin gives the electron to NADP plus, which is electron deficient, and here this is a reduction reaction. It gets reduced to NADPH, and there is an enzyme called NADP reductase. NADP reductase, which carries out this reduction reaction. It is known as NADP, and this NADPH, it is produced in the stroma. NADPH is always produced in the stroma. Now let us see reduction and oxidation reaction. Whenever a compound, this is a chemical compound, and whenever a compound it accepts electron, whenever a compound takes electron, it becomes reduced. Whenever a compound it accepts electron, it becomes reduced. Whenever a compound it gives electron, it becomes oxidized. Whenever a compound gives its electron to another compound, it becomes oxidized. Now let us see the electron transport chain. Electron transport chain, it consists of 3 electron carriers through which the electron passes. The electron loses its energy and moves. Whenever the electron passes from one electron carrier to another electron carrier, it loses its energy. So the electron, it moves downhill. The electron is said to move downhill because it loses its energy and moves. And the 3 electron carriers are plastoquinone, cytochrome B6F and plastocyanin. What are the 3 electron carriers? Plastoquinone, cytochrome B6F and plastocyanin. So electron transport chain, it consists of 3 electron carriers, and the electron, it moves downhill. Electron loses its energy and moves. And whenever it is moving from one electron carrier to another electron carrier, it loses its energy.